So we are going to continue with transcription. And remember that what is on the test is from where we left off last time. I believe it was lecture 7, right? So from that lecture until basically today, what I'll tell you, I am not going to ask you a lot on this lecture 14, okay? There are some points that are quite important that, of course, you should know, and I will emphasize those points. The problem now is that I don't remember exactly which questions I put on the test, but anyway, I will tell you where to focus, okay? So before I start, do you have any questions? Oh, everybody's good to go? Okay, yeah? Oh, the chapter numbers. I'll see after class. I always forget the chapter numbers. I think there is still a bit of chapter 3 on some protein, right? Yeah, well, like exactly where we stopped from last time until basically today. Part of chapter 3 on some protein. Yes, remember the prions? Yeah, those prions, remember what kind of diseases they cause? And how difficult it is to get rid of them? Basically, you cannot get rid of them, right? Yeah, I love to ask questions about prions. So there we go, happy birthday. Now, another thing that you really have to think about. I like to ask questions that ask you to actually use your brain, like your reasoning part of the brain. You have to figure things out. Okay, so you really have to understand the material in order to figure those things out. Okay, so for example, when we are talking about the experiments, remember that pneumococcal bacteria experiment? Yeah, when you had the live bacteria, there was the live bacteria, the good type that did not cause death. What was the name? What was the strain? There was a raw R strain. There was the S strain. Remember, the S is a bad S that causes death, right? And how it's shifting the information, how you get transformation. And then you had that experiment that was proving that DNA was the inherited material. You got to understand what those experiments did and what are the steps that proved that protein was not the genetic material. Carbohydrates were not genetic material. You have to understand what kind of enzymes were used and how they proved that protein, that DNA was the genetic material. Okay? All right, so let's go here. Continue. What's happening here with your RNA polymerase is all in this little bubble. So everything is stuck in there. It's not flying out and coming back. It's just there. So let's see here. Okay, so you start, you do your transcription, and then you're going to have to finish your transcription, right? So what happens at the end of the gene, so the gene was all here, so let's say you had here your promoter region, and then here is the gene. And then you have here some stretches that you're going to stop the transcription, okay? You don't really have a signal that says stop, okay? So what you have instead are some inverted repeat sequences that are going to cause the RNA, once it comes out, once the RNA is transcribed here, is going to cause the RNA to naturally fold into this hairpin type of structure, okay? Now, that hairpin formation is going to destabilize the RNA polymerase that is right there,
Okay, remember this bubble here is within the RNA polymerase, so the protein complex is not shown here, okay, for simplicity, right? So that hairpin structure destabilizes the attachment of that RNA polymerase to your DNA. And if you notice here at these repeat sequences, you're going to have a lot of adenines right here at your template strand, okay? So you're going to have a lot of adenines. When you are making your messenger RNA, you are going to add a lot of U's, right? Because U goes into RNA, not T, right? So we're going to have a lot of U's here. So the hydrogen bonds that are guiding that interaction, they are not as strong as if you would have CG type of hydrogen bonds, okay? So what happens here after the hairpin formation, your polymerase is going to detach from your DNA, and that's basically termination. Okay. So the way it happens here is that you don't really have a stop sign. You kind of have this road spikes ahead type of sign. Okay. And that, actually I have here those road spikes. Have you seen those? Like when you rent cars and you can only go one way, so you kind of like go the car this way. But the analogy here is that if you're driving your car on the way you're not supposed to, okay, you drive your car this way, what happens if you pass over those spikes? It totally destroys your tires. And then what happens to your car? You're driving and stop. Okay? So you didn't have to stop on the brakes, you didn't have to press on the brakes, it kind of like stopped naturally. Yeah. Okay, so that's the analogy there. By the way, one time I saw one person that actually did that, and I felt so sorry for that guy. I mean, they really, you could hear the tires going, oh, plow. Not a good sound. Anyway, but that's how you stop the transcription. Okay? So there are two types of transcription termination here in your prokaryotes. The first one is this type that just does this hairpin and then it dissociates, and that's the end of transcription. By the way, the end of the gene is somewhere before these sequences here. Okay? So this goes beyond the gene, and then there's a little more transcription, and then the polymerase finds these signals. Okay. So you call this type of termination row-independent termination. You don't see any extra protein here that is going there and prying open this interaction. So this is row-independent termination. So what you find there, you find those repeats of adenines, the strings of adenine repeats, at least 6 of them, and that is what is going to cause the hairpin loop, and then the things falls off. Okay, so this is a row-independent. So you can guess that the next one that you have is a row-dependent termination. So what is happening here? You have this little protein that is called Rho, okay? And what Rho does, it is going to interact with your messenger RNA, okay? It's going to find a sequence in your messenger RNA that it recognizes, and then it interacts with the messenger RNA, and it starts to climb towards your RNA polymerase. So this thing here is your RNA polymerase, okay? Now the way that this role works, it's like that exercise in the gym that you have that rope that comes from the ceiling. You know what I'm talking about? It's a kind of exercise that I could like never do, but some of you are apt enough to actually do that. You know, like you grab the rope and you pull yourself up on that rope. You know what I'm talking about? Difficult, right? But now imagine this guy is Rho and he's very good at climbing. Okay, so he gets the rope. The rope is your messenger RNA, nascent because it's being born, right? Messenger RNA. So Rho grabs onto the rope, to messenger RNA, and starts to climb it. But now Rho gets very annoyed because it is climbing the RNA at the same time that the RNA is being synthesized. So this rope keeps on getting longer, right? So it's already hard to climb, and you're climbing, and you're climbing, and then the rope is being fed from up there, and you're like, I'm climbing, and the rope keeps on coming, and you're like, I'm hurting. So the rope gets up there, and it's so angry that it dissociates that rope from whatever is coming from. Okay, it's like, that's the end of it. Throws rope down. It's like, what's going to do there? Oh, dissociates, it jumps down. Okay, everybody's happy. Okay. Rho is a helicase. This is how it can make this association of RNA and the DNA kind of like dissociate. Okay. So this is your Rho-dependent termination. Now, notice that when Rho gets up there, it is very close to that termination signal because look, you have a hairpin right there. Okay, so the hairpin also is forming with this complementary interaction of the nucleotides, but then after the hairpin, this is where Rho gets in there and it dissociates the RNA from the DNA. So you call that property of Rho, you call it RNA-DNA helicase property. Okay? So that's the story with the Rho and with the story of termination of transcription in bacteria. You got Rho independent, you got Rho dependent. If you see Rho, that's a Rho dependent. And now we'll get to the story of your eukaryotic transcription. Okay. So in analogy, you also have activation of transcription, initiation of transcription, elongation of your messenger RNA, and then termination. Again, with your eukaryotes, you also have some housekeeping genes, and those are transcribed constitutively, right? And this type of transcription is the one that you can actually control. You have a messenger RNA that, some of them are constitutively transcribed, and some of them you can increase transcription or decrease transcription. Okay, so what's going to happen here? First thing that you're going to do is actually to find the DNA. Okay, because remember, your DNA here, your gene is going to be associated with those histones, right? Remember, the histones are positively charged. So in order for you to reach your gene and actually be able to open up a bubble in your double helix, you're going to have to dissociate the histones, right? So what is the process that you do to the tail of the histone protein in order to dissociate it from the DNA? You neutralize the positive charge. You put acetyl groups on it, right? So you have those writers proteins, those are your acetylases. So you have your histone acetyltransferases. Those can also be called acetylases, right? They are going to put acetyl group on your histones. Okay. And now what's going to happen? Okay, you reached your DNA. You're going to have to open up the bubble. Okay, so here is showing your hats. Histone acetyltransferases are called hats, right? They go in there and they open up. They dissociate here your grabbing of those histones, and you can reach the DNA. So the analogy here is that if you want to eat those, candies that are like on the string, you kind of have to bite on the candy to reach the string, although nobody's interested in eating the string. But anyway, you get the idea. Okay. So how you free up the DNA from those associated proteins? So one way, you use those hats, right? After you're done transforming the gene, you're going to take out the acetyl group, so use your deacetylases. Okay. Besides that, you can also do chromatin remodeling. Okay, so this is a good test review, right? Chromatin remodeling that uses ATP. You have those complex proteins, of proteins that use ATP to push around this histone in order to reach the DNA. Okay, so you push those around and you find a piece of DNA that you can actually go to. Come on.
The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino-terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of two enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made.
Okay, so that's the story with that little movie. And here I put these slides showing one way that you can control gene expression when you actually need certain types of genes to be upregulated. So this is really modulation of gene expression. Okay? So modulation can be attenuation, which is repressing, or it can be activation, okay? Increasing gene expression. So what happens here? What do you see here? You see cell membrane. Okay, so these are your phospholipids in your cell membrane. And here you see a receptor on the cell membrane. Okay, this receptor is waiting for a signal to show up. That signal is going to tell the cell that it should upregulate certain genes. So the whole story with this is that this is a signal for interferon. Okay, so interferon is a molecule that could be considered a cytokine. It's important for immune system function. It's a molecule that is produced by all nucleated cells once they get infected by some virus. Okay, so let's draw here on the side. Okay, you have a nucleated cell that got infected by some virus. So here is like genetic material of the virus. Okay, so this cell kind of panicked and sent out alarm signals that those are interferon molecules. So let's say that interferon alpha or beta. Okay, these interferon molecules get in your bloodstream and it's going to diffuse and it's going to hit receptors of cells that are close by to this cell over here that got infected. Okay. Now, this cell that got infected, most likely it's going to die. It's infected, the virus is replicating, but the signal that it sends out is going to tell the other cells, like, do something, because there is a virus around and you are probably next, like, to get infected. Okay, so arm yourself because a virus is on the way. Okay, so that's the signal that is coming out of the cell. That's your interferon signal. So what's it going to do? This signal is going to go around here in the vicinity of that cell. It's going to hit this receptor here. Now this cell over here, okay, so this is the cytoplasm, and this is like, this is a membrane, so this is like the cytoplasm. Here is the nucleus of the cell that is not infected. Okay. So this cell over here has the receptor for interferon. So there we go. Interferon is going to bind to the receptor. The molecule of interferon itself is not going to go inside of the cell. It's going to cause some change in the conformation of this receptor. That is going to signal the inside cytoplasmic part of the receptor to change conformation and to do something. So the inside of the receptor is going to expose a signal that is going to start interaction with other molecules that already were inside of the cytoplasm. The molecules are just sitting there. Okay? Those molecules are like constitutively expressed. Okay? So what's going to happen is that those molecules are going to cause further changes in, you don't have to know all these details on what's happening with this receptor here, but something is happening. You had conformational change, you had the cytoplasmic part of this receptor that exposed some areas in a certain way. Now, this part is important for you. This area that got exposed because of the signal that it received, this area here got phosphorylated. So what is that residue over there? Which one is that amino acid? It has a Y. Tyrosine. Oh yeah, you don't have to know the letter abbreviations because I'll never ask you in the test. I think that's mean. But that guy is tyrosine. Do you remember the amino acids that can get phosphorylated? Tyrosine, serine. Which one is the third one? Huh? Threonine, is that it? Oh, I forgot, it has been too long. All right, so there are three of them, right? They have to have a hydroxyl group in order for them to get phosphorylated. If they don't have the hydroxyl group, they're not going to get phosphorylated. So there are only three of them with that, okay? This guy here is your tyrosine. So look at this. It got phosphorylated at tyrosine. Now, this thing is going to signal to some other molecule that is sitting over there in the cytoplasm, inactive. Look what happens. Pay attention to this because this is one of the thinking questions that you're going to get. Similar thinking question that you're going to get in your test. I promise you. So you got to know what's happening, okay? This guy here, once he got phosphorylated, it became activated as a kinase. What are kinases going to do? They put phosphates on something else, right? Kinases are going to put phosphate on something else. Phosphatases are going to take out phosphates from something else, right? So here I'm showing you the diagram of how it's happening in the cell. But if I were to draw what is happening, I would say something like this. Look, you have an enzyme here. Let's say this is your receptor. Okay, so here is a receptor that got phosphorylated. Okay, so what is the name of that enzyme that did the phosphorylation? Some kind of kinase. Kinase protein, right? So now your receptor got phosphorylated, and what is it going to do? It's going to act on something else that is a protein in the cytoplasm. And this kind is the STAT protein, okay, as example. Okay, so this is your STAT1 protein. And this guy that got phosphorylated is going to put a phosphate on your STAT protein. So what is the function of this guy once he got activated? It is working as a ASE at the end, is an enzyme that puts phosphates on something. It's a kinase. Itself, it is a kinase. So it was not activated. And here, okay, here it was not activated. It got a phosphate, it became activated. Now it can work as a kinase to get something else and put a phosphate on it. Okay? Now, let's say if this guy here, pay attention, this is tricky, okay? If this guy here acts, okay, let me make it easier. If this guy here acts on your original receptor to dephosphorylate it, it is going to inactivate your receptor. Because this guy here, once it is active, it can go over there and push this thing backwards. So what is the function of this protein over here? It's a, I mean, F is for, no, F is not for what you're thinking about, it's for the name of the protein. Phosphatase. It's a phosphatase, okay? So if it's taking a phosphate out and deactivating a receptor, so this guy here on its turn is going to be a phosphatase, okay? So you understand the diagram, okay? Are you going to have something similar in the test and you have to think about the function of the enzymes and what they are doing? Yeah, and what do you call this type of mechanism that, let's say, the last of a series of cascades, the last one came back and inactivated the first one? What do you call those types of mechanisms? Negative feedback inhibition. So that's a good test review, isn't it? Am I going to ask you about this? The answer is yes. Okay. So that's the overall idea of how you see this in a diagram and how you think about the action of those proteins. Is it difficult? No, because you just have to look at it and think, what is it the protein is doing? Oh, is one kinase, is it putting a phosphate? Yeah, it's a kinase. Is it activating the protein? Oh, okay, this goes from inactive to active. Then it does its job, and then this guy over here goes a feedback and is going to inactivate that. So this here must be a phosphatase. The phosphate is not there anymore. Okay, look at the picture and realize what's happened. Okay. Okay, so now let's get back here to our story of interferon, right? So interferon goes there, binds to the receptor. Interferon did not really get inside of the cell, it just bound to the outside. It causes all this change here to the cytoplasmic side of your receptor. Now, the cytoplasmic receptor here became a kinase. It phosphorylated some other proteins that were in the cytoplasm. So now, those proteins decided to hold hands, interact with each other, and once they are in this conformation, they expose certain sequences on their exposed site over here that allows interaction with other proteins that are part of the nuclear pore complex. So this thing here is your nuclear pore complex. And it's a really cool thing. It looks like a basketball hoop, okay? It has a little channel in the center and it has these little fibers that allow interaction like train tracks. It allows interactions of some transporting proteins that are going to bring this thing into the nucleus. Okay, so once this thing got activated, it dimerized and it gets imported. Look at that, how cool. It gets imported into the nucleus. You know how long it took me to do this animation? I wish there was AI at that time. Yeah. Okay. So now, what happened to that thing that got imported? Oh, this is another thing that is important for you to know. This thing here, that when it is in a monomer form floating out in the cytoplasm, this thing here out there is useless. But when it got phosphorylated, activated, dimerized, and imported, now it's a very important transcription factor. Transcription factor is going to do what? Interact with promoters. Okay, so this guy is going to find promoters on certain genes. Now, if you have to guess what kind of genes get upregulated through this process, what would you guess? So now you have to step back and say, what's happening over here, right? What's the story? Why did you activate this transcription factor in the first place? What was the signal? The signal was a distress signal from a cell that was infected. It sent a distress signal saying, watch out, we have viruses around here. So the stress signal is, You have to arm yourself because you're going to get infected. So this cell that got the signal, got the transcription factor into the nucleus, is going to find promoters of genes that have antiviral properties because it has to defend itself against a possible viral invasion. Okay? So the genes that are going to be upregulated, the genes are going to have promoters that this transcription factor is going to recognize. So you have transcription factor binding. You're going to have your RNA polymerase 2 binding over there to make your messenger RNA. Okay, so you have binding of your transcription factor. You're going to have, I don't think polymerase is showing there, but okay, more transcription factors. Okay, polymerase is not showing there for simplicity. And you're going to have here your transcription of messenger RNAs. That's what those red squiggly lines are. Okay, messenger RNAs, they're going to be exported from the nucleus. They're going to get out there in the cytoplasm and they're going to make proteins that are very important to fight the virus. Which virus? Whatever virus. Those are like general antiviral proteins. Okay? The cell doesn't know which virus is out there, but it knows that there are some viruses out there. So it's going to upregulate antiviral genes that are going to protect the cell from death because of the virus. Okay? All right, so that's the story with regulated transcription. So now we're going to go deeper into the story. Okay? So let's see what happens here. Okay. So you are going to find in the eukaryotic genes, you're going to find also those evolutionarily conserved sequences that are part of your promoters. Okay? The sequences, you call them consensus sequences. Okay, we have seen that before, so that's a quick review again. Okay, consensus sequences. Now, you're going to use those RNA polymerases. They're going to identify the consensus sequences. The first one that you find, oh, by coincidence, is the same one that is also present in prokaryotes, and that's your TATA box. The actual sequence of the nucleotides in the gene, look, it's like TATAAA. So that's really the TATA box. Okay, TATA. So here, a difference that you have between eukaryotes and prokaryotes is that this box show up a bit further upstream than you have your TATA box with your prokaryotes. So TATA showed up at minus 15, right? Remember, minus the negative sign indicates upstream before the gene. Okay, this TATA box here is like around minus 30. So minus 15 will be like around here. Okay, so your prokaryotes, your TATA box is going to be here closest to your gene transcription. Okay. So first thing you're going to see here is your TATA box. The second and third consensus sequences that you have, okay, is going to be here your CAT box. Okay, so this is okay, CAT box, right? So it's going to be more upstream. And you're also going to have another box that you call a GC box, even further upstream. So these are important consensus sequences that you find in the DNA. Okay. So a lot of promoters are going to have these consensus sequences because it's important for interaction of your transcription factors and guidance of polymerase to the site of transcription. Okay. Now, depending on the gene that needs to be regulated, you're going to have different repeats of each of those sequences and different combinations. Okay, so we have examples for that, like for example here. Okay, so in a gene here that you have, in this case, what is it? Okay, some gene here, you have TATA box. You have here GC box. You don't have the CAT here in this promoter, and then you have some enhancer sequences Now, enhancer sequences are going to interact with enhancer proteins. Okay, that's easy. So it depends on the gene, you're going to have different enhancers and they're going to have different sequences. It's very specific for the type of gene that you want to regulate, okay? Now look at this. This gene here is a gene that is down here, downstream of your promoter, is a gene that makes this enzyme that is called thymidine kinase. Okay, so the protein is thymidine kinase, the gene is over there. Look at the promoter of thymidine kinase. Kind of simple, okay? Kind of simple. And look, you don't have enhancer sequences here, but you always have a Tata box. This guy has the GC, but look, CAT is actually further upstream of your GC. So in general, GC goes down further, more upstream, but you can have different organization. The idea here is that each promoter, for each of the genes that you have to regulate, each promoter is unique on the expression, on the presence of those genes, of those repeat sequences. Those are consensus sequences, right? So each promoter is going to have their own repeat number and the own sequence of mix and match of those different consensus sequences. And so this is for your prokaryote. So talking more here about your specific polymerases of prokaryotes, right? Of eukaryotes. Prokaryotes, you have to worry about only one type of polymerase complex. That is your RNA polymerase, that's it. Prokaryotes, you're going to have at least 3 different types. I always have difficulty making that. Different types of polymerases, okay? 3 different RNA polymerases that you have to know. You have more, but you only worry about 3 of them, okay? And those are quite easy to remember, okay? First one you're going to see is RNA polymerase 1, okay? That RNA polymerase 1 is also called ribosomal RNA polymerase, okay? It's going to produce ribosomal RNA, okay? Now it's easy to remember that's RNA polymerase 1 because if you look here at the R, the lowercase r before your RNA, right? You have only one leg for that R, so that's your ribosomal polymerase 1, right? This RNA polymerase 1. So RNA polymerase 1 produces ribosomal RNA. Then let's look at the other type of RNA polymerase here. This is RNA polymerase 3. Okay. RNA polymerase 3 is going to produce tRNA. T3 starts with T, so that's your tRNA. RNA polymerase 3 also produces some ribosomal RNAs, but I'm happy if you remember that 3 produces tRNA. Okay? And then the other type that you have, of course, is RNA polymerase 2. This one is going to produce your messenger RNA. So this is the one that is going to be really, really regulated because you really have to regulate the messenger RNAs that get produced, right? How are you going to remember that polymerase 2 is for messenger RNA? Look at the M and you see 2 mountains. That's for 2 mountains. Okay, 2 hoops things. So that's your messenger RNA polymerase 2. Okay, so now let's see here what is important to know. Okay, so, I did not update this slide. You have about 25,000. We find lately that it's close to even 30,000 different genes that encode for proteins. Okay, so that's regulating those genes' expression is really the job for the RNA polymerase 2. Okay, so how do you know which gene, which messenger RNA to produce if RNA polymerase II is going to make all those messenger RNAs, right? So you have to really make use of transcription factors. The comparison between transcription factors of eukaryotes to the transcription factors for your prokaryotes is that for prokaryotes you have the sigma factors. So different sigma factors, those are your transcription factors that are going to coordinate gene expression. For the eukaryotes, you're going to have to identify what are the transcription factors based on cell function. Because keep in mind that all of your cells have the same DNA. Okay? All of your cells differentiated from the same fertilized egg. Okay? So there was one egg that divided, and then kept on dividing. You had cell differentiation, right? During the process of differentiation, you're going to silence certain genes, and you're going to upregulate others. The genes that you silence, those are going to be heavily methylated, right? The ones that get expressed are not going to be as methylated, as heavily methylated. So you're going to have different proteins that are going to be expressed. Some of those proteins are the transcription factors that are specific for that cell type. Okay? So if you look at cells in your liver, they are going to produce certain proteins. The only way they're going to produce the proteins is if they have the transcription factors for those genes. Compare those with cells on your skin, you're going to have other proteins being produced, and you're going to have different transcription factors. Okay? So you're only going to upregulate the genes in a cell type that you have the transcription factors for. Okay? Is that clear? Okay, so no transcription factor means no gene upregulation. Okay? Okay. So what are these transcription factors? Those are proteins, and they interact with the DNA, and they recruit other proteins to get over there and do the job. Okay? So two main classes of transcription factors. You have the ones that are used for constitutive expression, present in all the cells. Those are for your housekeeping genes, right? So you have your basal transcription factors, okay? Very conserved, all the cells are going to have that. And you're going to have low level of transcription, because those are always kept in check for housekeeping gene. And then you're going to have your other type of transcription factor, and those are your regulatory transcription factors. So those are the ones that are going to be present in specific cells that have to regulate certain genes. Now, usually, when you're talking about regulatory transcription factor, you talk about upregulating those genes. So those are positively regulated. So it means that you're not going to be producing those the gene products if you don't need the gene products. You're only going to produce them if you have the need for those products. Okay, so you don't waste energy if those gene products are not needed. So those are that's called positive regulation. What would be a negative regulation? If you're already producing the gene at a certain level and then you don't need as much, so you can shut down the production. That will be negative regulation. All right, so here are parts that get a bit complicated. There's a lot more to remember on what's going on here, okay? So first, you're going to have general transcription factors, okay? And you do have several subunits, so things that have to get together. Okay? So some TFs for transcription factors, they're going to interact with the DNA before your entire machinery gets there. Okay? Some other transcription factors are going to interact with the polymerase before it interacts with the rest of the stuff. Okay? Here. Come on. Okay. So here you see how your RNA polymerase 2 is going to get activated and start doing the job. First thing you need is your promoter region, right? Remember your data box is going to be around -25 of your start of transcription, right? That's going to be your data box, okay? And then you're going to have these transcription factors. A few of them you have to know. Not for this test, but I will push it to test number 3, okay? So you can start to get acquainted with them now, but you know, I'm not asking you about those like right away, okay? First one that you have to think about is this guy here, transcription factor 2D. So transcription factor for RNA polymerase 2, and this guy is the D, okay? Transcription factor D. What is going to do? D is just going to distort the double helix, okay? Yeah, it's funny, like as soon as I said it's not in this test, everybody's brain went like, oop, off state. No, turn it on because you're going to need it later. Okay. And it's a good review for interactions of transcription factors in DNA. Okay. So this guy is going to go and distort your DNA. It's going to open it up. And you're going to have a lot of complex that is going to join the party. Okay. So what is happening here? Look at this guy here. TF2D. Where is your TF2D? Is right here. It's a complex, okay? TF2D, it has some proteins. It's in a complex. One of them is this guy right here that is called TBP. The whole name of this thing is Tata binding protein. Guess where it's going to bind? The Tata box. All right. Tata binding protein is going to bind the Tata box, okay? Then what's going to happen? Once you bind your TF2D to your Tata box, you are going to bring 2 other transcription factors. You're going to bring 2A and 2B. So 2A is going to get in there and say, okay guys, just let's make sure that this interaction is stabilized. And then 2B is going to find the start site of your gene transcription. Okay? Then after that, you're going to have another factor that is right here, TF2F. TF2F is going to escort the polymerase 2, RNA polymerase 2, is going to escort this guy to this complex for interaction with the other transcription factors. Okay? So this guy here brought your polymerase right there. Then at this point, you're going to have another one, TF2H, right here, okay? That is going to get there, and H is for helicase. So H is going to open the transcription bubble that is going to allow for start of your transcription to take place right there. So lots of transcription factors here. You start with 2D, you go over here, and you get A and B. Then you get 2F that brings the polymerase, and then you get your helicase factors here that are going to make sure your start site is starting to do the transcription. Okay, so here, let's look here what happens. Okay, we did that. All right, so at this point here, you're going to have your transcription bubble inside polymerase 2, and you have a basal level of transcription. Okay, now here, more in the initiation, we already had that. Oh, this is a repeat. Oh, I didn't. You form your initiation complex. And here is showing to you what the Tata binding protein is doing. Okay, so this is your protein. Do you recognize pieces of the protein? You see here, what is this? Alpha helix. What is this stuff right there? Beta sheet, right? So you have different domains, and you're going to interact with the DNA and look at what the protein is doing. It is bending the DNA. So Tata binding protein is going to bend the DNA. By bending the DNA is going to open up a little loop over here, and that's the loop that is going to use for your transcription bubble. Okay, so binding does the bending. Okay, and there is more here to the story. Okay, more details on how transcription is actually starting here. So remember, you have your transcription factor 2D that comes with that binding protein. Binds over there. You bring A and B, they bind over there. And this big guy over here is your RNA polymerase 2. Okay, now pay attention what this thing looks like. It has a space over here that is going to be a transcription bubble. It has a tail. Okay, it has a tail. And it has these factors, these other proteins that are associated with it. So this thing is like, you know, a little turtle with a shell. Okay, it's a happy turtle. We got a big mouth here. It's a happy turtle. Okay, and it has a tail, and things are going to happen here. Okay, so what's going to happen here? You have your transcription factor 2H that is going to open up the DNA. So that's the helicase. You're going to start to make your RNA. Okay. Now look at this. This is the cool stuff that happens. Remember this guy here that is the helicase? It is going to do something else. It is going to put a phosphate group on the tail of your polymerase. Okay, lots of functions for this guy here. Now, once you have phosphate on the tail, you're going to change the conformation of that protein, and that's a signal for RNA polymerase 2 to start moving and doing its job. Okay, once it gets a phosphate on the tail, it's going to start doing the job. Okay, once it moves away from the promoter and starts to make RNA, you're going to have dissociation of the initial transcription factors, and you're going to have association of elongation factors because you need to stretch out, you need to elongate your messenger RNA. Okay, so these parts here, let's see what happens. Okay, so we stop here. You have your age that goes there, does the helicase. These association factors here, look, you are going to have, first here, you're going to have interaction with your DNA. Then you're going to have, oh, it's not showing exactly the age doing the phosphorylation, but you're going to have phosphorylation of the tail, okay? Once your phosphorylation happens, you start to make your RNA. And then other stuff is going to happen, okay? Okay. So this is how your transcription is starting. You can have enhancer regions that bind to enhancer proteins, and that can push also your polymerase to work. Okay. Okay. Oh, finally, you got some points out of this. So let me make sure here that you have your confidence level on. Go ahead, points. All right. So the question is, which of the following phosphorylates the C terminus of RNA polymerase 2? Yep. Yes. What I said, you know, these specific questions that are the specific pathways, it's not going to be in this. But generalized, let's say, you know, this is a good review for the test, then yeah. Basically, yeah. You're going to know the 3 different types of RNA polymerases. All right, so everybody done with this? What is your answer? C. Okay, here is your answer. That is your TF2H that does the phosphorylation. Okay. So there was another question, further clarification here. What do you need to know for this exam? You need to know the 3 different types of RNA polymerases, okay, of eukaryotic RNA polymerases, and what is it that they're transcribing, okay? All right. So here, what else you need to know here? Yes, I talk about enhancers, and let's see here, what else you need to know. Okay. All right, again, to emphasize here how it is that you are making your RNA, 5' to 3'. Yes, you need to know that. All polymerase is going to make 5' to 3' in this planet at least. Okay? All right, so here, your formation here of your RNA polymerase. This here is showing your transcription bubble. This guy is your RNA polymerase 2, okay? So this thing is going to transcribe, and then is going to come to a certain signal that there is no real clear termination signal. So it's going to keep on transcribing, and it knows that the messenger RNA is going to be processed anyway, right? So it doesn't really worry where it's going to end. But you do have certain signals that are similar to your prokaryotes. You're going to have signals for stretched out adenines, right? And you're still going to have hairpin loop formation of your messenger RNA, okay? And you're also going to have, the spikes on the road. Okay, it's going to you're going to weaken the interaction with the RNA polymerase. Same thing as we have seen before. All right. And here we go for a few slides on your RNA processing. Okay, so yeah, RNA processing is not going to be on your exam now, but it's going to be later for exam number 3. Okay, but you're going to have more time to digest this information. Okay, so what's going on here? First thing that's going to happen with that RNA that is coming out of the bubble is that you're going to have to protect it. Okay, protect your RNA. Protect it by putting some weird stuff at the 5' end. You're going to put a nucleotide with a strange linkage. This linkage is going to be from 5' to 5'. Okay? So 5' is hanging over here. You're going to attach a methylguanine to that 5'. So it's not just a guanine, it's a methylguanine. It means that you have a methyl group stuck to your guanine right there. Okay, methylguanine 5' to 5' type of connection. So that's the first thing that's going to happen. Okay, so this guanine here is methylated at the carbon 7 of your guanine molecule. Okay. Now, what is important to know here? Okay, so this is a strange place also for methylation of that G. Okay, once you have this 5' to 5' bond, you're going to attract a protein or a complex of proteins that is going to interact with that strange thing and is going to protect it. So that's going to be your cap binding protein complex. Okay, and why do you want to protect the 5' end of your messenger RNA? Why does it need protection? You have exonucleases in the nucleus. If that thing is not protected, it's going to be chopped off. Your RNA is going to be chopped up into little pieces at the same time that you're making it, so that's totally useless. Okay, so you have to protect the cap. First thing that happens, protect the 5' end. Okay, so those important things that you have to know for later. Okay. Okay, you also have methylation and your last nucleotide. Okay, so starting with the RNA processing, the first thing you're going to have when you're making your RNA is your pre-messenger RNA. Then that thing is going to be processed and is going to have a signal to leave the nucleus. if it is not processed properly, is not going to leave the nucleus. It means that if it is defective, it's going to be chopped up into little pieces, and the components are going to be recycled for other messenger RNA to be formed. Okay. 3 major modifications for your messenger RNA. The first one is your 5' cap. Then you're going to take out the intruder sequences of that messenger RNA. Remember, you have introns and exons. Exons are going to be translated, introns are going to be chopped off. Those are intruders, okay? So we're going to do splicing. Then you're going to put a tail at the 3' end. That's going to be polyadenylation. You're going to put a lot of adenines at the tail of that RNA, okay? All right, so now we're going to look again at those associated factors that are present with your RNA polymerase II. Okay, so let's see here. Okay, you can read that on your own. Here is what happens. Remember the tail that got phosphorylated of your RNA polymerase II? It doesn't only have the phosphates, it also has other associated proteins with it. Look at this. 3 different proteins associated with the tail. The first one here is something that has to do with capping. The second thing here has to do with splicing. The third thing here has to do with polyadenylation. So RNA polymerase II is carrying the proteins that are important for pre-mRNA processing. Comes in very handy. Okay, so what happens here? You are making your RNA, then the first of these proteins jumps into your nascent messenger RNA and allows capping. Then you're going to have, okay, this is a capping part, okay, and I already mentioned that, okay. So here are more details on how your capping works, okay. First thing you're going to do, you're going to remove a phosphate from your transcript. And after that, you are going to add that 5' to 5' guanine. There is a specific enzyme for that. The name of the enzyme is, thank goodness, guanyl transferase. You're going to transfer this guanine in this weird 5' to 5' bonding. Okay. After that, you're going to have the enzyme that's going to put a methyl group at the 7 position. So that's your methyl transferase. Okay. Oh, another question for you just before we leave. Which of the following enzymes adds GMP using 5' to 5' configuration? So this is your guanine monophosphate 5' to 5' configuration. I really have only one choice that makes sense. Person that answer E, please change your mind. Okay, do you need more time? No, everybody's good? Okay, what is your answer? B, that is your guanyl transferase. All right, so I'm going, oh, there's only one more, one more. Hold on, hold. One last slide. Technically speaking, you still have 6 minutes. I did put the battery in that lockable. All right, so one last thing about your poly-A tail, okay? The code for the tail, there's no code for the tail, okay? After your session of the DNA that has a lot of adenines, that is a consensus sequence that is close to the ending of that transcription, you are going to have a specific cleavage site. Remember, the RNA is going to be transcribed further down, like, I don't know, 1,000 of nucleotides down. There's a lot of thing here that is junk. You have to cut it off. So you're going to have a cleavage site. You're going to cut this rest of the RNA that is useless, and you're going to add a poly A tail to the 3' end of your messenger RNA. Okay? How big is the tail? Well, it depends. If it is a short tail, you're going to have 50 or so adenines that are added. If it is a long tail, you're going to have around 200 adenines that are added. Pay attention to this. I'm going to test you next time, right? The more adenines, the more stable your messenger RNA. Why? Because the bigger the tail, the more space you're going to have for some exonucleases to get in there and chop off the tail. You're still going to be functioning, okay? So the longer the tail, the more protected your messenger RNA is. How is it that the